Hello everyone, I'm here again to record a lecture video about the passive transport. So the first video that I had was about diffusion, which is the first type of the passive transport. Today, in this lecture, I will be discussing about the second type of transport, which is the osmosis. Okay, if you can still recall, they are of the same passive transport. Why? Because from high concentration to low concentration. But the difference is when we say osmosis, class, this is the diffusion of water molecules. Diffusion gani, di ba? High concentration to low concentration. But this time in osmosis, it is the movement of water. Okay, because in the diffusion, it is the movement of molecules, right? Whether it is a movement of solute. Okay, but this time, tubig ang transfer. Tubig ang move from one place to another. Okay, so does it need an energy? It does not need energy because this is from high to low concentration. So the same thing, always remember high concentration. It is always trying to reach solvent equilibrium. In the diffusion, it is the dynamic equilibrium. But this time, solvent equilibrium. Meaning si water ang ma-equal in both sides. Okay? First, in osmosis, there is the first thing that we need to know, and that is water potential. Okay? When we say water potential, class, that is the tendency, okay? That is the tendency of movement of water from one area to another. That is the mechanism, that is the term for moving of the water to one region to another. Okay, unsay kinahanglan na tumabaw-an or ma-understand regarding water potential. You can see in this diagram, okay, the first circle, water molecules is the small circles, right? And the solutes are the big circles. So you can see in this first circle, daghan ang tubig over the solute. Okay, in the second circle, you can see that there is an even distribution already of water molecules and solute. Okay, asa padung si water in this side? Okay? Okay, asa padung ang movement of water? First, we have to look at which one is high water potential and low water potential. Gi-encircle diri that this side has high water potential. Okay, nga naman, how do you say that it has high water potential? Because, remember, water potential is the movement nga mo balhin si tubig. So this circle, this region, we can say it has high water potential kay mubalhin man ang mga tubig diri to the other side. Okay, kay nga naman daghan man ang solutes diri. Okay, daghan man ang solutes. Ang tubig mubalhin pa dung sa region where there are plenty of concentration of solute molecules. Okay, in this other side, say that it has low water potential. Why? Because the water in this area will not move to the other side. That is why it has low water potential. It will not move to the other side because there are plenty of water molecules compared to the solute. Kay nga naman, kung magtanaw ka sa direction sa asa ang high water potential, okay, adto gyud na padung sa daghag solute molecules, class, because the solute molecules will suck in water. So yupon musuyop og water molecules kung daghag solutes. Meaning, kung daghag asukar diha nga area, musuyop na siya maayo sa daghang tubig, daghang water molecules. Remember, water potential will depend, no? The high water potential will depend to where to go. Padung siya dito sa daghan og solute molecules. So this one makes sense nga kaning nga area has high water potential because kaning mga circles nga gagmay move man na siya to this region, and this region has low water potential because this water molecules will not move and will remain. Okay, so again, that is the tendency of water to move from one area to another. So when you are going to look at the diagram, in determining asa ang high water potential or low water potential, always look to the region where there is plenty of solute molecules, because when there is plenty of solute molecules, gamay na siya water potential or low. Okay, I hope you see that. Okay, osmosis is the movement of water molecules class. Dili si solute ang move, but the movement of water molecules. So this is the first thing that we have to understand first, the concept of water potential. Okay, now let's look at this diagram. Okay. First beaker, okay? You can see this is semi-permeable. So the red are solute molecules and the blue circles are the water molecules. So after osmosis, you can see nga unsa man, what have you observed? You can observe that in the second diagram, you can see that nisaka ang water level. Nga nang nisaka man ang water level? Because water potential here is going. This area has high water potential. This area has low water potential. So the direction of water potential is going to the left or going to the right? Going to the right. That is why after osmosis, nisaka ang tubig. Why? Because ang direction of water potential here is going to the right because madaghan man siya og solute dinhi. Kay kung daghan og solute, okay, adto padung ang tubig. And that is osmosis, because osmosis is the movement of solvent. Okay, here. You can see, okay, in the normal osmosis, okay, what asa ang mas high water potential? Kani nga area this time. Low water potential, kani. Kay nga naman, gamay ra og solute diri. Kung gamay og solute, ang iyahang water potential taas. Right? Kung daghan ang solute, ang iyang water potential is low. Kay nga naman, si water move to where the region has plenty of solutes. Why? Solutes suck. Meaning, water will move to the area where there is greater solute concentration. Again, unsa ang ni-move? Ang solute ba ang ni-move? No, but the water are the one that is moving to the different region. So, think and understand this concept because we will apply this to the cell now. Okay? Again, high water potential this area, low water potential this area. If you're going to look here, high water potential here. Why? Because the water here will move to the other area where there are plenty of solutes. That is why this area is low water potential. Don't be confused about that one, huh? Because in analysis of the diagram, always look where there are plenty of solute molecules because that is where the water is going to, that area. Okay? So, in the examination, I will give you diagrams and I will ask where is the direction of the water potential and what region has high and low water potential. Okay? If we are going to compare diffusion versus osmosis, always remember, okay. Example, in this diagram, you can see this beaker on the left region, there are plenty of sugar molecules, right? So concentrated, ang iyang concentration gradient is taas sa left. So this is semi-permeable, meaning just like the cell membrane, it allows what goes in and what goes out. Okay, see, this one is lesser concentration gradient because there are lesser solutes that is present. Okay, so after the diffusion of water molecules, don't be confused that osmosis is diffusion of water molecules, meaning ang water ang ni-move. So right after osmosis, unsay mahitabo, ang water level ni saka diri sa left region. Why? Because always remember the solutes will suck in, meaning suyupon, musuyop og tubig. So, asa man ang high water potential? Ang left or ang right? Ang high water potential here is this side, the right side. Where is the direction? Going to the left. So, kani siya diri, okay, the left side has low water potential. So, asa padung si tubig? Going to the left. Okay, there are plenty of solutes here. So, expected that right after osmosis, this is what is going to happen. So, you can see that there is an increase in the water level here because ang mga solutes ni suyop sa tubig. So, high water potential, low water potential. Okay? So, and I will ask you this one during our synchronous session. I will give you diagrams that you will analyze water potential region and what is the how will it look like after osmosis. Okay, let's compare about diffusion and osmosis. Okay, if you will be given this diagram, what will happen after diffusion and what will happen after osmosis? Okay, this is the given. You can see here that there are more solutes in the right region compared to the left region. So where is the region that has high water potential? Left or right? Left, right? Because kaning tubiga muadto if we're going to think about osmosis. But in diffusion, okay, remember that it is not the solvent that will move, but it is the solute will move to attain equilibrium. So you can see here after diffusion, there is no changes of the water level, but the changes here is the amount of solute in every region. Because here, after diffusion, ang mga solutes ang nibalhin, right? Ni even both region on the left and on the right. But when we say osmosis, it's a different thing. Why? Because it is not the solute that will move, but it is the water that will move. Okay? So, when we say osmosis, this has high water potential, so the water will move to where there are plenty of solutes. So you can see that after osmosis, class, you can see that the water level is going up. Okay? It's going up. So, nisaka, it's not ang nisaka ang nibalhin dili ang solute, but it is the water. So the water potential is going to the right. And this is high water, low water potential. Movement of water is going to the right. Okay, why? Because of the presence of plenty of solutes on the right side, which sucks in the water molecules. Understood? Okay. Now let's apply this learning into practical life. We have this what we call as osmosis and food preservation. Have you ever if you make a pickle of foods or dried mangoes, marinate meat, how come they are not being spoiled if you are going to put sugar and salt? Okay? So, unsa may explanation ana? Nganong dili man madaot, nganong di man ma-spoil ang atuang food if we're going to make brine or example like this. Okay? I-brine nimo ang meat, di lagi siya madaot after pila ka days. Kaning mga sugar, pickled, dried fruits, di masad siya madaot. What is the explanation? Always remember that the cause of food spoilage is the bacteria and fungi. And this bacteria and fungi are unicellular. So, if the bacteria and fungi is surrounded by plenty of salt, plenty of sugar, which are the solutes, all the water content inside that bacteria will go out. Kay suyupon man sa gawas sa ilaha. So mamatay si bacteria, mamatay si fungi. Okay? So the water content inside the bacteria and inside the fungi will be outside their cell and they're unicellular. So dili maka-atraka og duol si bacteria sa meat na surrounded by plenty of salt, na surrounded by plenty of sugar. Okay? The same thing with dried fish. It will not be spoiled. Why? Because there are plenty of salts that you put into the fish. And the bacteria cannot go near that fish. Why? Mamatay man siya. Kaya nga nung mamatay siya, ang ihang water content inside sa iyang body, suyupon man sa mga asin. Diba? Kay living cell, manggihapon na siya. Another example is this prune feet fingers. If you are swimming, diba? Manglaba ka, mag-ingon ana, mag-crooked ang imuhang fingers or ang imuhang feet. Kung sa may explanation ana, ma'am, is nganong mo wrinkle man. Because water goes in. Dili ingon nga ni kuyos kay nanggawas ang imong ang water sa imuhang cell, but it is the other way around, which is the cell from the outside are going into your cells. That is why uneven nga nitambok, no? Because high concentration man ang water outside the environment of the cell in your swimming. Okay? So that is osmosis examples in daily life. Remember that osmosis is the diffusion of water molecules, meaning ang solvent ang move, and we have this water potential capacity. Okay, other subtopic of osmoticity, that is the strength of a solution in relation to osmosis. Okay, we have to understand the different types of tonicity: hypotonic, isotonic, and hypertonic. Okay, I will explain this one by one. Okay, because this is of medical use. No? First, we go to hypotonic. The first type of tonicity, we have this called hypotonic. When we say that the cell is under hypotonic solution, let's take a look at this diagram. Lower concentration of solute is outside the cell. High concentration of solute is inside the cell. Why? Because this is 20% salt. So meaning, mas higher siya kaysa 10% salt. So unsa may implication ani? What will happen to the movement of water? Remember that the solutes will suck in water. So if this is hypotonic, meaning mas taas ang concentration of solutes sulod sa cell. So what is the movement of the water? It will go, the direction of water potential is going into the cell. So what will happen to the cell? It will swell. It will mutambok, muburot si cell. Kay tanan nga tubig musud sa cell. Kay daghan kayong sulud sa cell. So ang water nga nasa gawas will go into the cell. And in return, what will happen? Is it deadly? Yes, it is deadly because the cell will swell and will burst open. And we call that cytolysis. Mu burst open, mu buto ang cell if you will be under this type of solution. And that is dangerous for the animal cell. How about for the plant cell? This is actually okay and good. Why? When the plant cell, you can see here, when the plant cell is with plenty of water in their central vacuole, if it's wilted, then you water it, tubigan mo kung naluya or nalaya. So what will happen into their plant cell is that we will have this turgor pressure. Turgor pressure is the pressure where the water is pushing the plasma membrane against its cell wall. So turgid na siya, mo turgid, mo barog, mo stay upright. So that is good actually for the plant. The hypotonic solution is actually good for the plants. Why? Because it will give them turgidity. Turgidity is very essential to keep them stand upright, because when they are standing upright, they have the maximum, optimum condition where they can receive the sunlight for their photosynthesis. Okay? Now let's move to the second tonicity, which is isotonic. Isotonic is the ideal condition of our cells. Why? Because iso is the same in both region, outside and inside the cell. And this is the best condition where our cells should be. Why? Because this is in the dynamic equilibrium, meaning there will be no changes for the size of the cell because freely moving in and out, kaya naka-dynamic equilibrium na siya. Okay, that is the best situation we need to attain. Third is hypertonic. When we say hypertonic, class, that is not good. Okay, looking into the diagram, you can see that the concentration of solute outside the cell and the concentration of the inside is lesser. Meaning, what will happen? What is the movement of water? So the high water potential now is this inside the cell, and the low water potential is the outside of the cell. So what is the direction of the movement of water? Move going out. Why? Because there mas daghan man ang solutes outside sa cell. So what will happen? For the cell in the plants, it will be plasmolysis. Meaning, this is the normal cell, but if it is under the condition of hypertonic, what will happen? It will shrink. So you can see here that the cell is detached from the cell wall. So we call that plasmolysis. But for the animal cell, that is another term, and we call that crenation. So the same thing that will happen, our red blood cells will shrink. So this is why this explains nga, if you are stranded in an island floating into the sea, would you drink the seawater or not to live longer? Knowing this, you should not drink the seawater. Kaya nga naman, mas dali man mang-shrink man imong mga cell. So mas dali ka mamatay. Mas dali ang dehydration because you have to avoid nga high concentration of solute ang gawas sa cell. Right? So the answer for that picture, you should not drink seawater because it will make the dehydration faster. The dehydration of the cell will be faster compared if you will not drink the seawater. Okay? So, summarize the tonicity in the effects of the effect of tonicity towards the cell. If it is hypertonic, class, remember, mugawas ang movement of water will be going outside the cell, and that will result to crenation and plasmolysis for the plant cell. If it is isotonic, very good. That is the best condition. Nothing will change in the concentration gradient. But when we say hypotonic, okay, that is still not good for the cells in the animal cells because it will burst, okay? It will burst, and that is not good. But for the plants, that is okay because it will give turgidity, no turgor pressure, and that is okay because they can stay upright. And that is what is osmosis is all about. Okay, starting from water potential, always remember that when there is plenty of solutes in that region, it will sucks in the water, and that is where the movement of the water will go through. And always remember diffusion, comparison of the diffusion and osmosis, what will happen after that, and the movement of the water molecules. And I hope you also know the explanation why our food won't get spoiled if we put sugar and salt. And learning tenacity, you know already the impact. If you will eat plenty of salt, like magkaon ka og mangga, uhawon din ka, magkaon ka parat, dahil kay chips, nga nung uhawon din ka, no? Because of that is hypertonic or hypertonic? Hypotonic, okay? So that explains the tonicity tonicity falls under the osmosis. Okay? So that is the second type of passive transport. I will proceed and I will finish to the last part of the passive transport, which is facilitated diffusion class, because this is short raman, no? Para mausa sa kaninga video. When we say facilitated diffusion, this falls under passive transport gihapon. Why movement of molecules is from high concentration to low. The only difference here is that naay ni facilitate nga mga integral proteins. Okay? So, passive transport is there are gatekeepers, there are gateways, no, nga nag-assist sa agianan sa molecules here. Okay? So, diffusion happens. Okay, this is a diffusion of solutes, meaning molecules na solid. Okay, and these are big molecules, okay, that are moving into and out of the cell. Okay, so when we say facilitated diffusion, we have this thing here, and these are the proteins. Either transport proteins, which are very specific to the type of molecules they will allow to get in. And there are also non-specific. Okay? So first, let's go to facilitated diffusion that are specific. Okay? When we say specific, meaning it will only allow unsay makaagi, no? So channel proteins are ang ilahang hitsura. What are their appearance is like a channel. That is why channel protein, because this is made of protein embedded into the cell membrane. Channel protein is very specific. So you can see pinacircle, and it will only allow, example, the sodium and chloride. When we say carrier protein, ang carrier protein, this is unique. Why? Because it will change the shape to unsa ang musulod na molecules. Also very specific. Okay, let's go first to channel proteins. I did mention already that the water cannot freely move into and out the cell, right? Because this is the fatty acid region, and this will repel the water. So how does water goes in and out? We have a gate. We have a gate embedded in our membrane, and we call that aquaporine. that is very specific for water. Okay, for water only. So, selective conduct water through them while preventing the passage of ions and other molecules. So does the aquaporine allow other ions to pass through? No, it's specific only for the water. So you can see, class, in our skin, there are plenty of aquaporins because we are sweating a lot. No, that is where we can see plenty of aquaporins. When we say carrier proteins, that is understood from facilitated diffusion, that is very specific because it will change, no? Ang iyahang agianan will change to what shape will go or will pass through. This is very unique because it is a shape shifter. Okay, example sa day mga diha ma'am. Sugars, amino acids, nucleosides, and uptake of glucose and transportation. Okay, so that is our facilitated diffusion. Remember, facilitated diffusion can either be channel proteins and carrier proteins. Okay, so next session, class, during our asynchronous, I hope you finish viewing this video, because in our synchronous session, there will be plenty of figure analysis, and there, of course, there will be sit work about passive transport in diffusion, osmosis, and facilitated diffusion. So, katong tulo, diffusion, osmosis, and tonicity, and facilitated diffusion, which are the carrier proteins in channel proteins. Okay, so I hope you understand everything, and I hope this will help. Thank you, and see you next video.